Yeah, the archaeal alpha diversity was.
My talk today is going to be microbiomes, probiotics, and health, and this is going to be a comparative metagenomics applications demonstration. I want to tell you that it contains very preliminary results, but I will take the chance just to talk to you about a couple of projects that I've been participating in for a few time. So this is my name. My name is Alejandro. I'm head of the sequencing unit bioinformatics in the National University of Mexico, Unidad de Secuenciación Masiva y Bioinformática. That's placed at the Institute of Biotechnology at the National University. And that's my email, just in case that any one of you want to contact me at some point. Or if you are in social networks, I'm very available in Twitter. So that's at Arroboso. So because I only have like 30 to 35 minutes, I want to start with the acknowledgements. There's basically two main projects that I want to thank people which were involved in them. The first one is the cheese microbiome, which is in charge of Doctora Mari Carmen Quirasco from National University as well in the chemistry faculty, and Doctora Alejandra Escobar Cepeda, who used to be my student first, then my postdoctoral fellow, and now she's working as a postdoctoral fellow, but at the EBI in Cambridge, in England. And also Doctora Mirna Olvera Garcia, who is working here in Mexico as well in the food industry. And well, the other project is the children gut microbiome, which is in charge of Doctora Ana Burguet and the Dr. Alfredo Laguna. Also, I would like to thank all the USMB members, which are the great people that I work with every day. They have been supporting. They have my back since 2012. I mean, 8 years ago, and all the organizers of this Latin American Students Council Symposium in this very odd year with the pandemic. And to everyone that is listening, I want to thank you as well. So moving to microbiota, microbiome, and metagenome. So maybe you have heard about these terms, and these terms, sometimes people use them indistinctively. But let's take microbiota against microbiome. So microbiota are microbial communities found in multicellular organisms regularly. They are the host of these communities. And also they include, these microbiota is composed, or it includes bacteria, archaea, protists, fungi, and viruses. Microbiota have been found to be crucial for immunologic, hormonal, and metabolic homeostasis of their host. And microbiome, in this case, refers to the collective genomes of these microorganisms. Therefore, metagenomes are all the genetic information of all the genomes in a microbiome. Can be used to determine the taxonomy of each community member. So, I mean, you can have the metagenome of a certain sample, and you can have the taxonomy of who lives there by several approaches. One, the most common will be the 16S amplicon sequencing, which is not properly metagenomics, but it's a taxonomic profiling. But in case you have a metagenome, which is it's taken from all the DNA, the total DNA from an environmental or a sample from a host. And you can use single copy gene markers to determine the taxonomy, which is way better than 16S. And obviously, I mean, because you have all the genetic information, it contains all the metabolic potential of this microbial community and how it's related to the environment. So the human microbiome, as you may know, it's influenced by several things. I mean, one of those is genetics, the environment, of course, diet, lifestyle, hormones, and sometimes the industry, all the products that we consume or use. I mean, it can go from the clothes to I don't know, a lotion, suntan cream or suntan lotion. They all affect, I mean, different parts of the microbiome. In this case, for example, if it's a lotion, I mean, it can alter the skin microbiome. And obviously, if you compare all of these tissues or organs, they have very different microbiome composition. So it's not the same bacteria that we have in the nose or in the mouth or lungs, stomach, etc. So, and they all have a specific role in maintaining the homeostasis of that specific tissue or organ. So, but also, I mean, as I said, I mean, environmental samples, I mean, very common. And I would like to talk about a very particular source of microbiomes, which is the fermented food. And in this case, I will talk about the cotija cheese. And cotija cheese is an artisanal cheese from Mexico. And it's very curious because it's a spontaneous fermentation starting with raw cow milk. And after 3 months of ripening period, I mean, the main question is, how come this cheese has no pathogens if you started with raw cow milk? I mean, there's no, pasteurization process before it. You start cheese. And also, you don't inoculate any bacterial started culture. It's completely on a spontaneous fermentation. I mean, with the bacteria that are present already in the milk, that probably it comes from the skin microbiome from the cow, also from the environment, sea salt from the Colima Sea, and also, I mean, other factors like the skin microbiome from the people that are the artisanal that it's buttering the curd. I mean, after you take the milk and just apply some proteases, yes, so you can start the aggregation process. And all of these factors are the things that we want to study to find out how this cheese has no pathogens in the end. So this is the work of Alejandra, and it's already published in Food Microbiology, and it's called Metagenomic Analysis of the Mexican Ripened Cheese Reveals a Unique Complex Microbiota. First of all, I mean, I want to tell you, I mean, that we sequenced this microbiome from a pool of 10 different samples from different regions in the Cotija regions. So Cotija region is it's in the mountains between the states of Jalisco and Michoacan. And we obtained around 226 million reads for a total of 22.6 gigabases of information. Obviously, from there, we removed all the Bose towers, the cow genome reads, that were not a massive amount of contamination. And after doing the assembly, I mean, we obtained a 104.9 megabases of assembly with a total number of contigs of 65,000 contigs, with an average length of 1.6 kilobases, being the longest contigs around 186 kilobases, and the shortest contigs is around 200 bases. Obviously, I mean, it's a very fragmented genome, but I'm going to explain later why. And the N50 and N90, which are metrics, I mean, to refer to how fragmented is an assembly, it shows that it's not too bad. I mean, half of the genome, we have them in several fragments where the shortest is the 3,854 bases. That's a very good space to search for genes. And in the case of the 1950, the N90, which represents 90% of the total assembly, is composed in contigs where the shortest is 547 bases. So it's a not too bad assembly, although it's fragmented. But the interesting part is that we have 143,000 genes that we can do several annotations on top of them. And obviously, I mean, the structured 16S ribosomal genes are also informative for the taxonomy. And in this work, I mean, we want to find both taxonomy and metabolic potential. So as you can see here in this tree, I mean, there's like 3 main domains where we can find firmicutes, protobacteria, and actinobacteria as the main groups for the community. Obviously, as I said, I mean, there's around 500 different geni that compose a very complex and unique diversity of a fermented food. So far, I mean, because we sequenced to a very deep coverage, I mean, we find pretty much most of the organisms that were present there at some point that are or were present. And let me tell you why we obtain a very fragmented assembly, and that's because, I mean, even though we have a lot of DNA information in this metagenome, I mean, in reality, there's only 3 species that are the majority of the DNA that you can find in the sample, and belong to three different bacteria, where you can see here. This is Leuconostoc mesenteroides, Busiella paramesenteroides, and Lactobacillus plantarum. And these three species represent more than 90% of the total DNA. But it's very good that all the rare biosphere that are composed by the other 500, or almost 500 genera. I mean, you have like a timeline where you can map back all the organisms that were involved in the fermentation process, because you have the evidence of their DNA still present in the cheese. Obviously, I mean, there must be some of these organisms that they are still available, but this is something we didn't check. I mean, we didn't check all the possible microorganisms, although Dr. Mari Carmen Quirasco has been isolating several of these bacteria, and I'm going to talk about that in a moment. Here you can see a metabolic map where you can find all the pathways and different pathways that are involved in the synthesis of different compounds that are related to the odor, the flavor, and different properties that gives the cheese this particular odor and flavor. I mean, whoever is Mexican and have tried this cheese, it has a very peculiar flavor and odor that, I mean, that it's not comparable, or is very unique in comparison to other cheeses around the world. In fact, this cheese won an award in Italy in a contest as one of the best cheese in the world in 2006. So after having all this information, trying to reconstruct the story of how this cheese was fermented after 3 months, I mean, obviously you started with an initial inoculum, which is, it comes, as I said, I mean, from probably the cow, the environment, the hands of the person that was in charge of buttering the curd. And it starts, I mean, like growing all the acid lactic bacteria. And in this case, because the lactic acid and the decrease of pH, I mean, is one of the mechanisms that the community uses to maintain a pathogen-free environment. Also, one of the things that we discovered is that at least we detected 3 different bacteriocins families. These are antimicrobial peptides. And the interesting part is that just some of the bacteria present in this cheese, they can produce the antimicrobial peptide. But all the community members, they have the immunity genes to be protected of these antimicrobial peptides. So in case that there's some pathogen that falls there, or any other bacteria that doesn't have these immunity genes, or I mean, they will die if they invade this community. So after 3 months, I mean, we have the lactic acid bacteria that is dominant, and in particular, the 3 species that I showed to you before. And because of this project, I mean, we were awarded by the National Prize for Science and Food and Food Technology. And another question that arose from this other work was that if this cheese, it can be a source of potential probiotics. And this is the work of Dr. Mirna Olvera Garcia, and she isolated and functional characterized the 2 strains of Enterococci that were isolated from this codica cheese, and how they were involved in the ripening. But in this case, let me show you how, after comparative genomics of these strains, we found out that they were very different if you compare them to free-living or pathogen strains. So this is a brief representation of the genomes. So we took a laboratory strain, a probiotic strain, one strain that comes from healthy infant. I mean, that it could be or not a probiotic, and also one isolated from an animal source, and a pathogen. So as you can see here, I mean, the strain isolated from cotija cheese is more similar to the probiotic than other strains that could be a pathogen. So in this case, pathogen is the most far away strain if you do the phylogenetics. And this is very, very refreshing, because that means that even if you hear the name of Enterococcus fecalis, I mean, it doesn't mean that it has anything dodgy there that is relating to feces. The same thing for Enterococcus physium. And so as you can see, also, I mean, the non-pathogenic strain is related to the one that we isolated from cotija cheese. And although, I mean, this non-pathogenic hasn't been proved, none of them hasn't been proved as a probiotic. There's a huge potential, I mean, of this strain to be probiotic. Obviously, I mean, it's very, it's not related to those pathogenic strains, although, I mean, we have to take in account or just bear in mind that even if these both strains lack genes related to cytolysins, addition proteins, aggregation substrates, and enterococcal surface proteins that are characteristic of the pathogenic ones, I mean, they possess antibiotic resistance. And even if this is not a trait of pathogenicity, I mean, you have to watch out, because if you use them after antibiotic treatment, I mean, they can overpopulate the gut, and this is not very good. Both strains were able to form non-structured biofilms, and a feature that could be correlated to capsule formation and potential probiotic behavior. And Mirna also tested in the lab that this strain of E. fecalis was able to adherate to mammal cells without damaging them. These were enterocytes in a culture, and I mean, this bacteria can get there and without damaging the culture. So that's a very good evidence that for the probiotic potential. Now, one of the questions that we have is, can we find bacteria from fermented food in the human gut microbiome? And let me tell you about a separate project that is related to the study of overweight or obese children in Mexico. And sorry, but the title was already in Spanish, and this is translated roughly as the childhood obesity as a consequence for the interaction between firmicutes and the consumptions of food with high energetic or caloric content. Okay, just making a long story short, in this study, they made this relation between the firmicutes and all the nutrients that were present in different diets, and obviously, the obese children they have a high they have a high calorie intake in their diets, and this correlates with this dysbiosis in the gut microbiome. Also, I mean, there's a second publication where, in very specific association between adiposity and cardiometabolic risk factors, we can find associated the abundance of Lactobacillus reuteri with the fructose intake. And we know that fructose is this syrup that it's present pretty much in anything that is sweet. And I mean, because it doesn't follow the same metabolic pathway as glucose, that it depends on insulin, it's very easy to ingest a lot of fructose without your body knowing about it. Okay. So so the question is, can we find the bacteria that we saw in different fermented food? Can we find them in the microbiome that they are present in children? And in this case, it's just a matter of or a proof of concept if we can establish how these fermented food, which in reality, I mean, contains bacteria that can be found anywhere, but it's just that in fermented food they find a better niche to thrive. If we can find them as well in normal weight children, that meaning that will point us to certain probiotic strains or species in comparison to the obese overweight, or in reverse, if we see some of these strains that are present in the normal weight children, but absent in the obese overweight. And obviously, if they are present in the normal weight children and in food or beverage that are fermented, that could give us a very good clue that they could be related to the maintenance or the homeostasis of the good microbiome, and they were probably present because of the ingestion of something similar to this fermented food, or maybe these fermented food are part of some of these children diets. So we have the diets, but in reality, we want to do this study as blind. As I said, I mean, so we have two groups of 24 and 24 children, obese and normal weight, respectively. And you can see that we start comparing different metagenomes and taxonomic profiles from different fermented food or beverages. For example, we have the kimchi, we have some cheeses, we have something called nunu, that it's the nunu, it's an African fermented food. And this is something very interesting about it, because as it was already published, in one of them, they found it that it's contaminated with E. coli, which is something that we don't see, for example, in other studies that we have done in fermented food, like in cotija cheese or even pulque, which is a beverage, it's a fermented beverage from aguamiel, that is this sap that is used for tequila as well. Obviously, we have yogurt, we have the fermented food metagenome from honey, for example, which is also another source of bacterial diversity that is very unique, as you may see in this heat map. But it's very interesting that, as I told you, I mean, E. coli is not present in any food except for the contaminated nunu sample that the authors already reported, and is present obviously in all the feces sample from the gut microbiome from the children, obese children study. But we have a variety of these strains that can be found at least one of these samples. And even if this is a very preliminary result, I mean, it's pointing us to the right direction to find or try to find probiotic samples. Obviously, I mean, we need to adjust and take this to the perfection in terms of methodology. But for example, I mean, here you can find that Enterococcus physium, it's present in several other fermented food, like soy sauce, for example, or tofu, and also it's present in the gut microbiome. But also, I mean, because we need to compare these strains at the genome level, we need to do the reconstruction of a single genome of these strains from each sample, and then compare it just to see if they share, just in the similar way that we did it for the cheese, cotija cheese strains, the physium on fecalis enterococci. As well, I mean, this is one of the many parts where that we need to study furtherly, and that can give us an insight of this relationship between well-being and health and probiotics and ingestion of fermented food. So the take-home message here is that any of the Latin America countries, I mean, we have fermented food that probably is worthy of studying at a metagenome level, just to find these strains that could have a metabolic potential that could be related as a probiotic in the human gut health. And also just very interesting, I mean, just how these communities produce also compounds that can be interesting from the biotechnology point of view, I mean, in the food industry. And there's a huge potential in this kind of study. I'm not sure how much time I still have left, but I think I have to leave some time for questions, right? So in case that I have some more time, I want to talk about some of the bioinformatics related to metagenomics. And as I said, as a brief background, I mean, we have been doing metagenomics or taxonomic profiling since long time ago. Basically, since humans started observing microorganisms in the microscope, but also, I mean, with the discovery from Carl Woese from the with the RNA ribosomal RNA gene as a marker for taxonomy, which, in my opinion, I mean, this is something that was really useful, but we don't need to use it anymore. Now we have complete genomes for metagenomes. That will be a huge improvement in the taxonomy as well. Also, I mean, the availability of taxonomic information has thrived or increased since the boom of sequencing technology. And this is just a graph we're showing, like the exponential growth of this information in the last couple of years. Well, in the last decade or 2 decades. Obviously, I mean, for example, it has been a great advantage to study, I mean, samples from the ocean or from other environments like the one presented today or in the previous talk. And so how do we do it? So there's 2 sequencing strategies that are important. One will be the target amplification or amplicons that they direct to a specific region of the 16S gene, in particular the B3 and B4. I mean, it's a very common and used region to do taxonomic profiling. But as I said, I mean, whole metagenome shotgun metagenomics is probably the best way to go in terms of study microbial communities. And this is why we decided to evaluate different strategies for taxonomic annotation using both approaches. So in this case, I mean, we don't want to point out our finger to any of these methods because they all work. And basically, it depends on the taxonomic level that you want to report, but basically they all work. But the important thing about this work that I did with as well with Alejandro Escobar is that at least you need to know what is the error rate that you will find depending on the combination of the software, the database, and the sequencing technology or sequencing approach that you decided to use. So what are the variables and how they affect the results? Basically, we started with simulated reads from 840 prokaryotic reference genomes in 3 independent libraries. So we evaluated, for example, I mean, 16S RNA methods like CHIME or SPINGO, also Metaxa2 and parallel method, which are these 2 methods can be used for both approaches, for whole metagenome shotgun or 16S. And specific ones for whole metagenome shotgun is Metaplan2, MOCAT, Kraken, and Clark. Some of them, they are based on sequence alignment, and the other, like the ones on the bottom, are based on K-mer spectra. So the ones that are based on whole metagenome shotgun databases, they rely on single copy marker genes, and the K-mer spectra, they rely on RevSec genomes. Also, I mean, we tested for the ones that we were testing for 16S databases. We tested several databases for this information, like RDP, Silva, Green Genes, and Metaxa2 database. That is a very small but very well curated database that comes with Metaxa2 software. So what we did was a confusion matrix where we evaluated the true positive, false negative, and true negative, and from there we evaluated each database and each approach in different combinations. So we evaluated the coverage versus errors per sequence plots is the one that I showed you just a few seconds ago. We evaluated sensitivity, specificity, accuracy, and the Matthews correlation coefficient, which we think that it's a very good metric to evaluate methods. And this is because it considers all results from a confusion matrix. So all these true positive, true negative, false positive, false negative combinations, they are well weighted in these Matthews correlation coefficient. And you can use it pretty much to evaluate any method where you can calculate these values. And here, for example, you see that at pretty much family level, they perform, acceptable in an acceptable way. But if you want to refer to genus or species, probably the best for them are CHIME and parallel meta with the Metaxa database, proving that a very well and small curated database improves your results. And also, I mean, species and subspecies level, when you're working with 16S ribosomal genes or amplicons, is not a good idea. It's not going to be reliable. So just take care about this. As I said, I mean, this is the list of you can use. So for example, using Chime or Metaxa with green genes as well, I mean, doesn't work that bad if you're at family level. But I mean, please don't use it for species or strain level. But conversely, if you work with whole genome shotgun data, pretty much they perform really well, even at the species or subspecies level. So this one, for example, methylplan2 is probably the best, even for subspecies level. And this is one that I recommend personally. Although it might not be the best one, it was the best one in 2018 when we run this benchmark. So concluding remarks about the methods. So if you go to class level, any method will work. And if you want to go below class level, for example, order, family, or genus, I mean, BLAST-based methods, which are the sequence comparison methods using BLAST with small databases, I mean, this is the way to go. And for, as I said, for species and subspecies, it's not recommended to use 16S. I mean, it's not that it's not going to give you a result, but the result is not going to be as reliable as with other approaches, as the whole metagenome shotgun. Any method will do it, and in the particular case of species of subspecies, single copy marker genes are the best methods to do a taxonomic profiling at this taxonomic level. And so in case you want to reproduce our methodology, you can find them in this GitHub repository. You can find all the instructions for the benchmark and all the data sets, and that's pretty much it. So I think that was the last one. Well, here I want just to point out, I mean, how the whole genome shotgun works. Basically, you can take environmental samples or samples from anywhere and just have different approaches. As I said, single copy marker genes is like the best way to do taxonomic or phylogenetic abundance profile. You have several methods for this. I'm not gonna go into this in very detail, but also, I mean, the assembly is a very good approach. I mean, just to do not only the taxonomic profiling, but all the metabolic potential studies that I showed to you before. And there's in particular one new method that is called the High-C library, that in this case, just before extracting your DNA, you cross-link with formaldehyde, all the DNA inside the cells before extracting the DNA, and that will give you fragments that are physically related to each other inside the genome. And this will improve your genome assembly or metagenome assembly, and will allow you to reconstruct whole genomes from metagenomes, or what is called metagenomes assembly genomes, MAGs. Probably you will find this term in some publications. So with that, I will leave some time for questions, and thank you for listening.
Dear Dr. Alejandro, it's an amazing talk. Thanks for sharing this a lot of knowledge benchmarking tool. This is maybe a first question about the fermentation. How is the best tool to do everything for bioinformatics from metagenomics? This is a very useful guide. We proceed to some question. We have some question here with great talk, Alejandro from Camila. SHAMI2 sometimes reports at the spatial level. Do you recommend to ignore this prediction altogether when reports your research?
Well, I mean, it's not that you have to avoid them or ignore this prediction. It's that you have to be very careful and take them with a pinch of salt, because you know that there's a high probability that you will have some errors at this taxonomic level. Although, if you check your results against the literature or any other source of information that can validate the biological role or the environmental role that you're trying to demonstrate or to assess, I mean, that will be a good result.
All right. Thanks. We have another question. And how do you think that this scientific knowledge will be transferred to artisan cheese producer?
This is a very
These
good
are
question. This is a very good question, and in fact, I mean, this is one of the main drives for this study. And the thing is that because I mean, we cannot export this cheese because it doesn't. For example, the FDA rules. I mean, it will never pass the FDA rules in the States. And worst of it, there's several other cheeses that are similar to cotija cheese that actually are fake cotija cheeses, but they are made from not from raw milk, but processed milk. But still, they get pathogens like Listeria or Salmonella. And so what we want to do is just to hand these to the artisans and give them like a profile on how an innocuous safe cheese looks like at the microbial level. So if you can take this at any sample and you have these 500 or something, well, this in this case will be some most of the 500 genera that we characterize. I mean, they must be present in any cheese because actually it came from a pool of sample of these cheese from different producers, and we believe that this has to be present in all the samples from any cheese in the Cotija region. So this is how we can help them, just giving them this profile. We haven't figured out, I mean, how to present this evidence of the FDA, but the sole paper, the sole publication can work as a proof of concept or a proof of safetiness for this product.
all right. Thanks. We have another question. Hi, Alejandro. Great talk. This is Jessica from Colombia.
Yeah, okay.
Regarding the microbiome study in artisan cheese, you think that your results are potentially relevant to assist in improving cheese quality and safety?
Yeah, indeed. Yeah, I mean, yeah, this is one of the objectives. I mean, how we can start like doing artificial consortium that can be used for cheese fermentation. That will be one option. Or maybe you can use or add some of these bacteria to other products or isolate them so you can also produce certain compounds that can be used as odor or flavor compounds in other products. I mean, there is a very good potential in these terms.
We have another. In the characterizations of potential probiotics, is there a measure of statistical criteria to determine probiosis when it evaluates the group of genes related to adhesion, acid resistant, production of vitamins, etc., or it remains on presence absence?
As I said before, I mean, just take my results as a preliminary and with a pinch of salt, because I mean, we haven't done this yet. I mean, like properly. So you're right. I mean, the absence and presence was one of the first approaches, very naive approach, just to see. I mean, if we can find things that you found in the food, in the gut microbiota of these children. And we expected to see a difference, but actually we didn't see any of them. But it was very interesting that, I mean, the comparison between different fermented food, I mean, they all were different. I mean, for example, some of them came from dairy product fermentation, others from vegetables, others from cereal, and they all share some bacteria that are related to the fermentation process. But we don't know the specific role of each of them in each food. So one of the hypotheses behind this is that if you can find this in fermented food, you just take these profiles as a representative source of what you can find anywhere in any other food, even if it's not fermented. And if these bacteria can be present in the gut microbiome, that means that either you're ingesting this kind of food, I mean, like regularly, or there's a process where you maintain them, or they can remain in your gut, I mean, as probiotic in the case of those children that were not obese, and probably they were absent in the microbiomes of the children that they were obese. So these are the questions that we want to solve, and we're on the way of doing it. Obviously, we need to figure out what are the best like metrics or statistics to relate not only the genes, but also the whole genomes. I mean, with the synteny, and also all the nutrients that are present in the diets of these children.
All right. Thanks. We have time for another question. This great talk, Alejandro. Is there any sequencing technology more used for shotgun metagenomics, or these are not relevant?
Well, right now, just because the time that we have this technology being around is Illumina. Illumina sequencing, either for amplicon or whole metagenome shotgun sequencing, it will be more used or more available. And in fact, all of the data that I presented was from Illumina, from public repository that data. It was sequenced with Illumina technology. But also, I mean, metagenomics used with nanopore are already available, although they're not as abundant or as frequent as this one from Illumina. But they have different advantages. One of the things that is one of the things that you have to evaluate are the biases present in all these technologies. In the case of Illumina, because you have to amplify the DNA at some point, either in the sequencing process or before in the library process, preparing process, I mean, you will have bias amplification, which is not too bad in the whole metagenome shotgun, but is not absent. While in the nanopore sequencing, because you are doing a single molecule sequencing, you're not amplifying and you don't have these biases. But I think right now we don't have all the software tuned to use this information, and also it's more expensive and it's more low throughput, or it's not as high throughput as the Illumina one, and this is why we haven't used it that much in terms of metagenomics. But if you ask to me, I will advise to do Illumina sequencing in a whole metagenome shotgun library, in addition to a high-C library that will allow you to resolve hundreds of complete genomes. And we have done this in other projects, and it's working really well.
Right. Thanks, Dr. Alejandro. We have no more questions, but this a very interesting question always about how technology used to from metagenomics or whole genome sequencing. Maybe this a mix together with Illumina and nanopore. This another solution, depend of your goal of investigators. Well, this maybe this end of the long day, with close with good knowledge, and actually maybe we have some words from the committee organizing teams, announce awards. Maybe Camila talk a lot more about the awards from poster and oral presentation.
Camila, just, I would like to ask you if you're gonna share or you're sharing the presentation. Sorry, are you compiling all the presentations, but for sharing them with the rest of the people?
So we are recording it right now, but I don't know if it's going to be open. I think that's something we are going to talk about later.
No, because I can share with you my presentation, but I'll probably send it to Camila.
Okay.
By the way, sorry because I wasn't replying your emails, but it's just that they get lost in a big pile of emails that I have. But I'll send to you the link for the presentation in case you want to share them with the rest of the people.